So what I thought I'd do today is to share with you how we're approaching understanding why children get disease, particularly malformations, and how we might not only learn the causes of those such defects, but also use that knowledge for potential therapeutics as they get older, and also for regenerative medicine. And the story I'll tell you will relate specifically to our efforts in the heart, but at the outset I'd like to state that the approach that I'll share with you and the method through which we're going about this can be applied to your favorite organ. Whatever disease you're studying, I think the principles that I'll share with you can apply to that, and I'll try to come back to that point repeatedly. And I think you will. And so the way I think about how we'll approach medicine in the future is that there are revolutions going on in several different areas right now and have really been transformative over the last 4 to 5 years. And those have to do with genomics and genetics and stem cell biology. And it's the marrying of the power of those technologies together, as I'll show you, that I think is going to open the door to really finally get past the way we approach many aspects of medicine right now, which is to simply address the consequences but not the root of the cause, and that's what we'll try to get to. And so our efforts have really stemmed from children with heart malformations and trying to understand why it is that children have malformed hearts in the hopes, as I mentioned earlier, that we might be able to, in the far future, prevent those, but in the nearer term, at least try to address the afterbirth, the sequelae of those defects. And I'll come back to that point in a moment. For many congenital heart defects, we may not be able to change the natural course of that in utero defect because that's already happened very, very early during development. But particularly at a place like UCLA that's had such a long and storied history with adults with congenital heart disease, you all know that it's not over once you're born and once we do the surgeries. There are long-term issues that are a consequence of the early defect that occurred in utero. And maybe we'll be able to address some of those down the road. Now, it's been our hope that if we understood how the heart normally forms, that we might be able to get to this question. But we've also, for a long time, realized that if we understood how nature normally makes a heart in utero, we might be able to leverage that information for adult forms of heart disease, be it heart failure that occurs after heart attacks or heart failure that occurs in our congenital heart population as they get older. And right now, at the end stage, the only real treatment we have for heart failure is a transplant, and I know you all do a lot of those here. But ideally, if we could harness the knowledge of how nature normally makes a heart and use that to create new myocytes or regenerate a damaged heart, maybe at least in some cases we could avoid the transplant route through regenerative medicine. And I'll come back to that at the end of the talk. So our laboratory, our efforts can be broken up into 3 distinct themes. The first is to simply understand what the gene networks are that are dictating early embryonic cell to adopt a cardiac fate. What are the gene networks that are instructing that cell, and how might we be able to leverage that? And we've made significant progress in that area over the last 2 decades. And that has allowed us then to ask, are those gene networks the ones that are disrupted in the setting of human disease? So I'll share with you some of our efforts on understanding the human genetics of congenital heart disease and how we might use stem cells to address mechanisms in drug discovery. And then finally, the third aspect that you heard earlier about our lab is to use that knowledge of developmental biology to reprogram and change the fate of cells in the adult heart into new muscle cells. And so the story really starts by going back and using a variety of different animal model systems to understand how the heart forms in utero. And what I've depicted on this slide are the series of morphogenetic steps that we know occur in the mammalian heart as the heart is forming. And we know that, I won't go through the details of the developmental aspects in this slide, but just want to point out that we now know that there are distinct pools of progenitor cells as the embryo is forming that will contribute to unique parts of the heart. So the red cells that I've depicted here, initially at 2 weeks of gestation, very, very early, start to align themselves in this crescent shape that you see here. And those cells, the red cells, are fated to become the left ventricle of the heart as this crescent shape fuses in the midline to form this heart tube that starts beating by 3 weeks of gestation. As this tube loops, the red cells, you can see here, are really fairly specific to the left ventricle only. The yellow cells that you see here contribute mostly to the right ventricle and the outflow tract of the heart. And then there are these blue cells that contribute, that are neural crest derived, that contribute to the vessels that emerge from the heart. The reason this is important is that this begins to form a basis for us to think about congenital heart defects that might affect one part of the organ and not the other. So we know that in congenital heart disease, the whole heart doesn't not form. Obviously, then that would be embryonic lethal. But only specific regions are disrupted, and it's likely because we're disrupting specific pools of progenitors as the heart is forming, and the rest of the progenitors are fine. And what that means is that we really have to dissect the molecular networks that control those individual pools of progenitors to really be able to get at this understanding. Now, using fruit flies, fish, mice, and chick embryos, we've been able to learn a lot about the genes that control each of these progenitors and each of these steps. But some of the earliest cell fate decisions that occur at this early stage when the cell is first being told to become a muscle cell have been very hard to get at in animal models because it's happening within hours. And so for that, we had turned to the embryonic stem cell or pluripotent stem cell system in a dish where we can, on an hourly basis, take cells out of this dish as they're turning into heart cells. And what we've found is that we can fairly accurately now map a pluripotent stem cell in a dish and track it as it becomes either a mesodermal or ectodermal progenitor. And we now know that these mesodermal progenitors will form either these red or yellow cardiac progenitors that I showed you in the previous slide, so we can parse those. And we also know that these yellow progenitors in particular are multipotent in that they can turn into not just muscle cells that beat in a dish, but also the endocardial cells and endothelial cells of the heart and the smooth muscle cells. And so we've been able to, it's turned out that the same genes that are important in an animal that we've mapped out are the ones that are dictating cell fate even in this system in a dish in vitro. And together, by studying using both of these systems and going back and forth between them, we in the field have been able to map a series of genes that control each of these different pools of progenitors. And some of those genes are shown on this slide. The names aren't important, but what I want to illustrate for you is that we can assemble these genes now in sort of a hierarchy, and we know who's controlling what to some degree, and I've tried to illustrate that. And this is knowledge, at least from my lab, has been contributed by a number of talented trainees over the years, many of whom are running their own labs, including Hiro Yamagishi and Vidhu Garg, who are both pediatric cardiologists and running their own laboratories now. So it turns out that as we studied this process, we did find that many of these genes that you see on this slide are in fact the ones that are mutated in the setting of human disease. But in every one of those cases, only one copy of those genes were mutated and the other copy was normal. So in the worst case scenario, we're only dialing down the dosage of these networks by 50%. And in some cases, they're point mutations that are hypomorph, so the protein's still there, it's still functioning, just not as well. So maybe we've only lost 30 or 40% of that protein's activity, yet that's still enough to cause human disease. And what that told us is that the dosage of these factors was extremely important for the cell to control properly. And this was this realization was back in the early 2000s. And right around that time in 2001, a whole new area of biology had been described in lower organisms, in fruit flies and in worms. And in 2001, it was shown for the first time that there was these tiny RNAs called microRNAs that actually were present also in humans. And the reason I tell you about this is these microRNAs, which are only about 20 to 22 nucleotides in length, so they're really, really small, they serve to titrate the dose of protein made by a cell. And the way they do that, they're encoded in the genome, so they're transcribed from the genome like a regular gene. There are about 1,000 of these encoded in the genome. They come out to the cytoplasm, and they actually physically interact with messenger RNAs to negatively regulate how much protein is made from that messenger RNA. So you can think of it as a dial to control the dosage of protein made from messenger RNAs transcribed by the genome. And so it occurred to us, well, if these are present in the human, maybe these are involved in controlling that dosage, and we should go find if there are any that are present in the heart. We didn't know of any and what their biology might do in mammals. And so it turns out that we've now identified many, and the field has overall has found that there are microRNAs in every cell type in our body. This particular one that I show you here was the first we studied, and it's called microRNA 1. These are all numbered, 1 to 1,000, if you will. And this microRNA is very specifically active in muscle cells, striated muscle. So what you see here is an embryo where blue indicates where this microRNA is active, particularly in the heart, which in this embryo is right here, and these stripes here, which are the precursors to skeletal muscle in the somites. So this is you think of this as a muscle-specific microRNA, and we've published a body of literature on this microRNA that I won't go into today, but the net effect of this knowledge we gained is that this microRNA plays an integral role in telling an early progenitor to become a striated muscle cell, and it actually shuts down the smooth muscle gene program, and it's one that locks down the fact that a muscle cell should form and stay as a striated muscle. And this microRNA turns out to, as we now know for other microRNAs, is deeply embedded in the major genes that I showed you earlier that control cardiogenesis, and it's regulated by those same DNA binding factors that turn genes on and off, and it feedbacks to regulate those in a complex network. Net effect titrating the dosage of these key pathways that I mentioned are disrupted in human disease. Over the years, we found that each cell type in the heart has its own unique microRNAs that function in very much the same way to either dictate that cell fate or lock it down into its fate, or help that cell respond to various stresses that might be impinged on that cell or organ. And some of those are shown here. Again, the names aren't important, but I wanted to illustrate that each cell type has its own cell type specific microRNAs that titrate the dosage of the key pathways. And so by using the genes that are sort of master regulators that turn other genes on and off, as well as the genes that titrate dosage, the microRNAs, together we've been able to place these into, and as well as signaling proteins, place them into stem cells in a dish and quite efficiently direct a pluripotent stem cell down a path to create fairly pure cultures of either cardiomyocytes or endothelial cells or smooth muscle cells for various purposes, either using these cells for regenerative medicine or to use them to try to understand why human disease occurs, because we can do this with human cells in a dish. And this is just an example of what these cells might look like if we take these pluripotent cells and differentiate them in a dish into beating cells. They look something like this, where they form a syncytium and they beat in synchrony. Now, there are embryonic stem cells that we can do this with, human embryonic stem cells that are pluripotent. But there's a new type of pluripotent cell called induced pluripotent stem cells that have really opened up our ability to do things we couldn't do before related to human disease. And I'll tell you about that in a moment. But first, one of the roadblocks we had in trying to understand human disease, of the genes that are shown here that have asterisks that we and others have identified are mutated in human disease, is that we could find the genetic mutation in humans, and now with ability to genome sequence, we can find a lot of variants in people with disease. But if that's all you know, that this gene causes this disease, but you can't figure out why, you're not going to get to a way to intervene and create a new therapy. What you need to create a new therapy is to understand the mechanism by which that genetic variant actually causes a problem. To do that, you need to have cells in front of you to study. And we've used animal models for many years to try it as a surrogate to try to do that. But I'll tell you, for most congenital heart defects, and it's true for other birth defects, when the human mutations are heterozygous, and you go make that heterozygous mutation in a mouse, the mouse doesn't care. It's fine. And if you make a homozygous mutation of a really important gene, they die very early as embryos, and that doesn't really recapitulate the disease process. So for most of these genes that are shown here, we've done that and haven't learned so much. Our lab in particular had identified through human genetic studies mutations in this gene here called Notch, and I'll come back to that in a moment, that was heterozygous, as well as a really important gene called GATA4 that caused holes in the heart. And we didn't learn a lot from the mice that we made, unfortunately. So the real breakthrough came with the discovery from one of my colleagues that I had recruited at Gladstone, Shinya Yamanaka, in about now 8 or 9 years ago, where he found a way to take a skin cell, or now a blood cell from a human, and convert that adult cell all the way back so that it behaved just like a human embryonic stem cell. And it turns out that a combination of just 4 factors that were really important, they're DNA binding proteins that turn on and off genes all over the genome, those 4 factors were enough to take an adult cell and turn it into what he called an induced pluripotent stem cell, or iPS cell, and I'll use that term again. So iPS cells basically are an adult cell that's been converted to a pluripotent stem cell. But the beauty of this now is that you have in front of you a stem cell from that person that didn't involve making a human embryo, so it doesn't have any ethical issues around it. And those cells are genetically identical to the patient, so you can imagine the use of those cells for transplantation for regenerative medicine that wouldn't be rejected because they're genetically identical. But for the purposes that I'll share with you today, the other really major part of this advance is that you can now take somebody with a genetic variant that causes disease, and if, let's say, it's heart disease or brain diseases, normally you'd want to study their heart or brain cells, but obviously you can't take biopsies of those to study. But now you can make these stem cells from that patient with a genetic variant, turn them into the cell type you want, and then you have in front of you cells from that patient with disease that you can use both for studying mechanisms and drug discovery if you want to try to correct that disease process in those cells from that patient. And I'll show you an example of how we've done exactly that here in the recent past. So conceptually, I'm going to show you a little movie to show you what this means conceptually. So we've always thought that these pluripotent cells you can imagine as cells that sit atop of a valley, and as they differentiate and to turn into specific cells, they roll down a hill and go into a certain valley that might represent, each of these valleys representing a different cell type. And the energy to go back up that hill and reverse that trend would be too great. So once they've gone down a valley, they're fixed there in the adult, and they could never go back down up towards a pluripotent state. And so if you imagine these balls rolling down this hill, the iPS discovery really was showing that there was a combination of master regulators that you could put into a cell that would push that ball back up the hill and restore its ability to now become any one of these different cell types again. And we never thought that an adult cell had that potential. We thought it was locked down in those valleys. And so this was such a fundamental discovery and changed conceptually how we thought about the plasticity of an adult cell that it was awarded the Nobel Prize in Physiology and Medicine in a short 5 or 6 years. And this is Shinya, the day he won the prize in Stockholm. And it's probably one of the shortest periods between discovery and actually awarding this, because it truly changed the landscape of stem cell biology and how we thought about cells in general very, very quickly. So with Shinya's lab down the hall, we quickly made iPS cells from these patients that our lab had described with genetic mutations in these various genes that I mentioned to you earlier. And I'm just going to share with you briefly the highlights of a discovery we've made around this Notch1 gene, which is a very important transcription factor that controls development of actually most organs in your body as it's forming. But for some reason, if you have a heterozygous mutation in this gene, only the heart is affected, and only the aortic valve is affected, and not the rest of the heart. And we had come to this through a genetic study 10 years ago, but as I mentioned, we had gotten stuck until very recently because of this issue with not being able to model the disease. And so the work I'm going to share with you was published just a few months ago by a very talented MD PhD student in my laboratory, Christina Theodoris, who fortunately wants to go into pediatrics and is in back in her third year of medical school now. And she used this IPS model to finally tackle why is it that patients with this single gene mutation get aortic valve disease. So I'll share with you highlights of that right now. So the disease that we had found Notch1 is involved in is calcific aortic valve disease. But while this disease often occurs in the adult, its basis is in a congenital malformation. It's a very common form of adult heart disease, and it's an age-dependent disease, the calcification part is. But a majority of these individuals are born with bicuspid aortic valves. So instead of having 3 valve leaflets, they only have 2. And actually, as a congenital malformation is actually the most common congenital malformation. 1 to 2% of the population worldwide is born with the bicuspid aortic valve. It can cause a severe problem in the neonatal period with a thick valve that needs to be balloon dilated, or it can be subclinical and people don't even know they have it till they get older, and then it starts to calcify. So why this valve that's bicuspid is predisposed to calcifying, we've never understood. But as I mentioned, 10 years ago, we had come across this large family shown here, where there are 10 or 11 people affected by this disease. And it ranged from the full spectrum of this family, from an infant who needed to have intervention to a 50-year-old who didn't know they had disease until they got a calcified valve and had to have it surgically replaced. All in one family, which ties the two, including hypoplastic left heart syndrome, which is the most severe form of aortic valve disease, where the aortic valve doesn't even open, and as a result, children are born without a left ventricle. So in this family, when we scanned the whole genome, it turned out that a point mutation in Notch1, heterozygous point mutation, was enough to cause the disease. We did learn through studies in mice that the key cell type involved was the endothelial lining of the aortic valve, but we didn't know why that caused it. And so Christina made iPS cells from 4 or 5 family members that had the disease and 4 or 5 that didn't. She took those iPS cells and she did maybe the most deep interrogation of those cells that has been done to date, by seeing where Notch binds to DNA to see what it turns on and off, what the epigenetic landscape is with and without the mutation, and how genes are altered. And she did this in the presence or absence of a laminar shear stress. So endothelial cells and the aortic valve are exposed to shear stress. So as the valve opens and closes, all this blood is flowing past it. And we knew from clinical studies that the side of the aortic valve that's exposed to laminar shear stress is actually protected from calcification. That side doesn't calcify. The back side of the valve that doesn't have that shear stress is the one that calcifies. So something about that shear stress protects the valve from calcifying, but we didn't know how that occurred. And I think Christina has figured that out through the studies. So I'm just going without going into the details, give you the punchline of what she found. And that is that Notch, which I haven't told you, is a protein that acts in the nucleus to control genes turning on and off, but initially it actually sits at the cell membrane, as depicted here. And because it sits on the cell membrane of endothelial cells, it's in a position to serve as a sensor, if you will, of that blood flow going across the cell. And that, in fact, is what Christina found, is that Notch seems to be a key sensor of this shear stress and gets activated, goes to the nucleus, where its job is to turn on a whole program to suppress the calcification program. So that's its normal job in these cells, is to keep the calcification program down, and specifically to keep a bone gene program suppressed. So osteogenesis. And what happens if you don't have enough Notch is that this suppression of the bone gene program is relieved, and now the endothelial cell begins to think that it should be a bone cell. It changes its fate, if you will. And now all these bone genes are turned on. There's also a variety of inflammatory signals that get activated. So she not only found that Notch was the key sensor for this process and key regulator, but she also, through doing a lot of gene network analysis, made the remarkable finding that there are about 1,000 genes that were dysregulated, even if you just dial down this Notch protein by half. But those 1,000 genes, of those, 80% of them were controlled by just three key factors that served as the nodes that were then turning on and off these other 1,000 genes. The reason that's important is that if you want to correct these 1,000 genes, it would be really hard to do that with a drug if you had to correct all of those. But if it centers down to a couple of key nodes, well, then maybe it's feasible to get a drug that could actually affect just a few key nodes, and those then reach out and correct everything else. And so based on that finding, she proceeded to do a drug screen with these iPS-derived endothelial cells in a dish from these patients, and asked, could I screen a few thousand drugs that actually in this case have been FDA approved, so we know could be used in people, and might we be able to find a drug that corrects this disrupted gene network? And so I'm just going to show you two slides that illustrate actually that we've been able to do that. And this is I'm going to simplify what you're seeing here to a few points. So this is a principal, what we call a principal component analysis. But the point is that we're using each of these dots, the position of it illustrates what state it's in, if you will. It's where its genes are. So in these blue dots are what a normal person's endothelial cell would look like, and you can see they all clustered sort of in this area together. The green cells are where the Notch heterozygous patient cells might cluster, and they're separate from the blue. They don't really mix. That's the point. Now, all these red cells are when we've tested each of these few thousand drugs on the patient cells, and most of those red cells cluster with these green cells. That means they're not really doing anything. The drug doesn't really affect those cells. Those mutant cells are still just like the regular mutant cells, even when you give them drug. But a few of these, shown here, this one, this one, this one, are mutant cells, but when exposed to that drug, they now cluster and hang out with the regular normal cells. That analysis then shows us that a single drug, each of these single drugs, is actually moving this mutant cell gene network close to a normal wild type. So these now have become, we have about 10 of these, and now we're going back and testing these in vivo in a model of aortic valve disease, and we think that these could potentially be drugs to prevent the progression of this calcification. So the story here, what I've carried you through is our initial human genetics, use of human genetics to find a human mutation that causes disease, making iPS cells to find the mechanism of disease, and then using those cells to find new drug candidates that can be tested. And I'm very hopeful if any of these work in our in vivo models that these are already ready for a clinical trial because they've already been tested in people before. So that illustrates sort of the approach and path to go from a human disease gene to a new drug. And you could do this for your favorite disease. Okay, so for the last part of the talk, I want to turn to a different theme in the laboratory, which has to do with regenerative medicine. So what I've told you so far really is about genetics and disease modeling and drug discovery. And I'd like to turn now to how can we use the knowledge that I've described for regenerative medicine purposes. So there are a variety of reasons for heart failure. In the adult, it's largely ischemic heart disease. In our kids population, it's if they have a right ventricle that's been systemic, that reliably fails as they reach their second, third decades of life. Regardless of the cause, the bottom line with heart failure is that we don't have enough heart muscle cells. And you can take conditions like this or others and say, okay, if we don't have enough heart muscle cells, let's try to take a stem cell like I showed you before that's beating in a dish, and let's try to inject that into this area and get those cells to take up residence there and help contraction of the heart. And we're trying to do that, as are many other people, but it's been difficult. When we inject those cells, the vast majority die. Those that persist fail to electrically integrate with their neighbors and contribute to pumping. And I think we can overcome those over time, but right now those are major hurdles. A few years ago, we used the observation that I'll tell you in a moment to try to take a different approach. And that is that the heart, even though it looks like it's all muscle, is only half muscle. The other half of the cells in the human adult heart are fibroblasts. They're support cells for the muscle. They contribute to the scar that you see here after injury, but they're never meant to be muscle, and they have a completely separate embryonic origin. But recognizing that those cells are already there, they're sitting in the organ. And I've told you before now that we can take a fibroblast and turn it into a stem cell and convert its fate. We asked the question, might we be able to harness this vast pool of cells that are already in the organ and not turn them into stem cells, but ask them to become new heart cells? So the question is shown here, could we take these fibroblast cells that are already in the organ and reprogram them directly into new muscle cells right there in the organ, not take cells out and put them in, but use those cells that are already there and turn them into new muscle cells? And this was the work that Masaki Ieda, a very talented postdoc, who's also trained as a cardiologist and has his own lab back in Japan, asked several years ago. And to do this, we basically said, okay, we know a lot about how nature builds a heart. We know now the key gene networks that are dictating the cell fate in an embryo. Could we leverage that and basically throw the kitchen sink at an adult cardiac fibroblast and ask, is any combination of those genes enough to induce this conversion? And so it turned out, Masaki screened about 20 transcription factors, 20 microRNAs, and it turned out that a combination of just 3 key developmental factors were enough to take a cardiac fibroblast and push it towards what he called an induced cardiomyocyte, so more like a cardiac muscle cell. And these 3 factors, one is GATA4 that I mentioned we had shown earlier is a human disease gene. Another is TBX5, which actually is also a human disease gene that causes Holt-Oram syndrome. And we had previously shown that these 2 proteins actually physically interact and regulate genes. And then a third gene, MEF2, which is also a very, very important cardiac developmental regulator. And so Li Qian, another postdoc who now has her own laboratory at University of North Carolina, asked, could these 3 genes that Masaki showed worked in vitro in a dish, could they also do the trick in vivo, which is what we really wanted to do, and convert the cells that are already in the organ into new muscle? So to do this, she used a genetic model where we genetically labeled the fibroblast cells in the heart with a green marker, a green fluorescent protein, and then ligated a coronary artery, injected directly into the muscle with the gene therapy approach, these 3 genes, and then waited a few weeks and took the hearts out. And we can section these and look at what happened. We can also digest these into individual cells in a dish and ask, do we now have green cells that used to be fibroblasts that now look like muscle cells and beat? And it turns out that this process turned out to be actually much better in vivo than it even was in a dish. And I'll show you a movie here. So this cell, of these two cells, this cell is not green and basically represents a pre-existing muscle cell that was already there. This cell is green, which means it used to be a non-muscle cell, and now it's been converted into a different kind of cell. And this cell, when we electrically stimulate, as you can see here, beats just like the endogenous cardiomyocyte. So this represents a newly born cardiac muscle cell. And so going back to this valley diagram I showed you earlier, this is a bit different conceptually. We did a number of experiments to ask, does that, as these cells switch their fate, do they ever go through a pluripotent or progenitor state? And it turns out they don't. They go directly from one adult cell type to another adult cell type without ever going through a stem or progenitor cell state. And so what's really happening here is that by introducing these three factors into a cell that's already in one valley, it's fixed there, that instead of going up the hill, what basically is happening is these cells are jumping over from one valley to another and going from one cell type to another without really going back up the hill. So it's conceptually different, and over the last several years, many other investigators at our place and others have shown that you can do something similar for brain cells, for pancreatic cells, for liver cells, et cetera. Okay, so what we know now is that when we induce this conversion in the heart, these cells that are newly born actually electrically couple with their neighbors, which is important because that has to happen for them to increase output. They're actually electrically very similar to adult ventricular cells, and they improve cardiac output when we measure it by MRI, which is the most rigorous way to measure that. By histology, it looks something like this. This is a control heart. The left ventricle is here. This is a scar that occurs after an infarct. And here you can still see that there's still thinning of the ventricular wall, but now interspersed in the scar area are threads of muscle. And we can use that same color tracer to ask, are these threads in fact newly born? And in fact they are. These are all newly born muscle cells that used to be non-muscle cells. And this is what an MRI of these hearts would look like, either in diastole or systole in the control setting or with these factors. And you can see that in this setting that there's less blood here during systole, which indicates the increase in cardiac output, and we can quantify that here with this dashed line indicating what the normal cardiac output should be in a mouse. The blue bar is what it is after infarct in the control setting, and here green you can see it's not normal yet, but it's a whole lot better, the cardiac output with these factors. So obviously we don't want to treat heart failure in a mouse. We want to do this in human cells. So we immediately put these three factors into human cardiac fibroblasts in a dish, and we're very disappointed that they basically did nothing. So we're clearly missing something. The human process was more complicated. So Zhidong Fu, another postdoc who now has his own laboratory at Case Western, went back and screened for 20 other factors to see what we might be missing, and he found that if we added 2 more factors to these 3, then that was enough. So then these 2 were another important developmental regulator called MESP, and then a nuclear hormone receptor shown here, that together now these 5 factors in a dish could do about as well as what we could do with mouse cells in a dish. So knowing what we knew, even though it was imperfect, that in mice, that in vivo, the reprogramming was a lot better than what we could do in a dish and this on plastic in a non-native environment, he pushed forward with these 5 factors and asked, could this work in vivo? And we obviously can't do that in humans right away, so we went to the next best thing, which is a pig model, whose heart's about the same size as the human heart, the physiology is very similar, and those 3 factors also don't work in pigs. So Palmer Yu, another postdoc, then led the work in pigs that I'll show you here towards the end. So in the pig, what we do is we create an infarct with the balloon occlusion of the left anterior descending artery. And then we wait 3 days and measure by MRI, measure where the cardiac output is and how big the scar is. That gives us a baseline of how much damage we've done. A couple of days later, we come in with an endoscopic device that we've created just for this project that allows us to directly visualize the damaged muscle and inject with the gene therapy approach, again, these 5 genes, 5 factors. And then we wait about 2 months and then do MRI again and histology to see if we're able to create new muscle and if we can improve cardiac output. So this is what a pig heart looks like. This is the apex of the heart, and you can see here, I hope you can appreciate it's a bit blanched compared to here. And so this is the area of infarct here, and this is where we inject the different factors. So if we, after 2 months, if we cut a section through here to see, do we create new muscle, we don't have the elegant genetic labeling tools we have in mice to tell if this used to be a non-muscle cell. So we've done the next best thing, and I'll show you that here. So I mentioned earlier that the 3 genes that worked in mice don't really work in pigs, so we use that as a control. We injected those 3 genes with a color marker that would indicate that a cell that got infected is red. And the strategy we used was such that only the fibroblasts would get infected. So only fibroblasts would be red, or non-muscle cells. And so if we ever saw a red cell that looked like a muscle cell, it should have come from a non-muscle source, if that's clear. So here in the control setting, even with these 3 factors, if we look, stain these heart sections with a muscle marker shown in green, we see these beautiful sarcomeres, which are the contractile units. And then you see a red cell here, which is fibroblast. We never really saw a cell that looked like a muscle cell with these green stripes that was also red, because muscle cells don't ever get infected. In contrast, with the addition of these 2 factors and co-injection with this red marker, now you can appreciate here, I think, all these cells and these that are both red and green, meaning they used to be non-muscle cells, and now they have these beautiful sarcomeres and are cardiomyocytes. So after looking at about a dozen pigs and with a bunch of controls, we think that these 5 factors might be the right combination to induce this cell fate switch, even in a large animal that's closer to humans. So in the last 6 months or so, we've moved forward with this approach to actually ask, can we do it well enough and get enough cells to make a difference in cardiac output? And just in the last few months, we have begun to get that data, and I'll show you that here in the last, I think, 2 slides. So we're very encouraged by this result. We're doing more numbers to make sure that this is reproducible in a large number, but the initial results are very encouraging. So what I'm showing you in this slide is the change in cardiac output in these pigs over time. And so if we start off here in a normal pig, and I mentioned after we create an infarct here, if we measure by MRI their cardiac output decline by day 3, the red line indicates the treated pigs and the black is the control pigs, and they both decline about the same amount. And in the control pigs, that decline over the next 2 months continues to get a little bit worse, so their cardiac output is down here. And in contrast, after delivering these genes in the experimental pigs, we see now not a decline, but rather an increase in the cardiac output in these. And I think we're very encouraged by this because we have not optimized the delivery system in this as yet. I think we can do much better by targeting more cells, but even in this relatively crude delivery fashion, we're already getting an improvement in cardiac output, even in this large heart. So we're very, very encouraged by this, and we've also looked by MRI at the scar size. And what happens normally in a pig is that the scar size between day 3 and at 8 weeks is that the scar undergoes normal contraction and it shrinks a bit. That's normal. About a 20 to 25% shrinkage of the scar is what's normal, happens in any scar, any infarct. In contrast with these pigs that we've treated, the scar shrinkage that we've observed is about 60%. And that correlates with the improved function and the creation of new muscle. So by a number of parameters, we think that with this cocktail of factors, we're able to convert enough cells to actually make a difference. And now we're doing a larger number of pigs and doing a number of safety studies to make sure that this is safe. And there are a variety of reasons why we might run into problems with that as we go forward, but we're at least encouraged that we've got a preclinical model that might be might work. So at the moment, we're trying to refine delivery by testing a number of different viral vectors to deliver. We're also testing a number of chemicals that might either enhance the reprogramming, and we've found several that actually make the reprogramming more efficient, and maybe even an all-chemical approach that can replace the gene therapy, but we're in the middle of those studies. And what we're also encouraged by is that this paradigm of harnessing cells in an organ that's been damaged and converting those to the cell type that's been lost is not unique to the heart, but almost every organ has such support cells around that can be leveraged to convert for regenerative medicine purposes. And in fact, a number of other investigators at our place and others are taking this approach now for a variety of other cell types and diseases. And actually, the very first clinical trial with this approach has already started with hair cells in the ear for hearing loss, because obviously the safety issues there are simpler, and you only need to convert a small number. And so Novartis has started a trial earlier this year with a gene therapy approach with a single gene that can convert support cells in the ear into hair cells, and that's underway. So this part of the story, really, what I've told you, goes from simply understanding the developmental biology of an organ system and the gene networks that are important, to leveraging that gene network to convert a cell's fate, all the way to going to a preclinical large animal model that might actually get us into a clinical trial in the not so distant future. So that approach, I think, again, can be applied to virtually any organ, and currently is being applied there. So I'm going to close there and leave a few minutes for questions. This is the lab that has done all the work that I mentioned. They're a fabulous group of scientists and physician scientists who are very, very dedicated, brilliant folks that I have the privilege of working with. A number of former members I had tried to mention as I went along, and I've had great collaborators also at Gladstone and outside of Gladstone. And I'd be happy to take any questions. Thank you very much.